Bismillahirrahmanirrahim. Let's continue with process of cellular respiration, a cellular process which generates lots of ATP from starting molecule glucose. And previously we talked about how glycolysis is the first step of cellular respiration, which takes a glucose molecule, C6H12O6 molecule, and converts it into two pyruvate molecules. This process occurs in the cytosol of the cell. In other words, if you were to draw a eukaryotic cell, here's a eukaryotic cell with its nucleus. Let me try to do a better job here. So here's a eukaryotic cell with its nucleus, okay? And if you were to draw a mitochondria over here, which is of interest to us, particularly at the moment. So glycolysis is a process that occurs over here, and glycolysis generates two molecules of pyruvate. Now there are two molecules of pyruvate, and the pyruvate is going to journey into the mitochondria, and the remainder of the processes of cellular respiration are going to occur in the mitochondria. So the first process, the glycolysis, occurs in the cytosol, whereas the remainder of the cellular respiration processes are going to occur in the mitochondria. So this is very important. Let's just highlight this in the cytosol. So now the pyruvate is going to make its way into the mitochondria, which I shall try to draw now. Okay. Now, if you recall, mitochondria has is a double membrane structure. It has an outer membrane and an inner membrane, and the inside of the mitochondria is called the mitochondrial matrix. Mitochondrial matrix. Okay, so pyruvate is going to make its way into the mitochondrial matrix, and let's draw a pyruvate again, and this is the carboxylic acid end of the pyruvate over there. Okay, now over here, the pyruvate is actually going to be converted into a molecule which is reasonably famous in itself, and that's called acetyl-CoA. Now, acetyl-CoA, I'm going to draw this out first, and then I'm going to explain to you what happens in this reaction. And you can observe that this part of the pyruvate doesn't change. The change actually occurs over here. So what's going to happen is that this carboxylic acid group is going to be released from the pyruvate in this reaction. So this carboxylic acid is going to be released, and it's going to generate carbon dioxide molecule like this. Okay, and this enzyme, this reaction is catalyzed by an enzyme. So this is an enzyme. So it's catalyzed by an enzyme, and the enzyme requires a coenzyme. Coenzyme is like a Batman-Robin type of relationship. It's a helper of an enzyme, so that's called CoA, and hence the CoA, so coenzyme A. So coenzyme A is kind of like a helper of the enzyme. So pyruvate combines with CoA to form the acetyl-CoA, and the original pyruvate part of the molecule is right here, and that's going to remain in the acetyl-CoA like this, and the coenzyme group is going to be over here. So basically, this is a reaction that's going to deliver the pyruvate molecule into the next step. So the first step of cellular respiration was glycolysis, and the second step here is going to be conversion of pyruvate into acetyl-CoA. Okay, now this is going to generate for us some more NADH. Okay, which is, as we said, it's an electron carrier, so it's an electron carrier or taxi, and that's important for us. So NADH is an electron carrier. So the second step is this conversion, and we generate NADH from this, and then carbon dioxide is released. Okay, and this CO2 molecule is this molecule from the pyruvate over here. Okay, this carboxylic acid group. Now, what's going to happen? This is acetyl-CoA now is going to join in with into a cycle, famously known as the Kreb cycle, also sometimes known as the citric acid cycle. Okay. Now, if you recall from our previous discussion, a cycle is a series of reactions which start with certain substrates and it changed into different compounds, and the end result, however, is the very starting substrate. So a cycle is a series of reactions which result in the production or regeneration of the original substrate, in this case A. So what happens here then is that acetyl-CoA joins the cycle, which is known as the Kreb cycle, and with a series of reactions, okay, acetyl-CoA is going to be regenerated, it's going to be left up like this, okay? So that's the Kreb cycle, okay? And what's going to happen during the course of the Kreb cycle is that the carbon dioxides are the further processing of the acetyl-CoA occur, and carbon dioxide is going to be released. So another 2 molecules of carbon dioxide will result. So it's important to understand that 3 molecules of CO2 are released in total from here, but then you have to understand there were 2 pyruvates, correct? So a total of 6 carbon dioxides are released, which makes sense because glucose is a 6-carbon molecule. But that's a waste product of the Kreb cycle. What the Kreb cycle really gives us is it gives us ATP, which is the purpose of cellular respiration, is to give us energy, right? So it gives us ATP, and it gives us more electron-rich molecules. It gives us a lot NADH, okay, and it gives us lots of NADH. And it also gives us another electron carrier, which is FADH2. Now, these are electron carriers. Okay, so what does the Kreb cycle do? It processes acetyl-CoA and generates for us energy-rich molecules, ATP. So where's ATP generated here? ATP is generated over there, and ATP is generated here in glycolysis, and I must say there's another ATP molecule, ATP-like molecules, GTP, which is also a cousin of ATP, so to speak, which is also an energy-rich compound that's generated. So ATP is generated over here, and GTP is generated, and ATP is generated over here. Now, one last thing I must suggest here, and acetyl-CoA basically delivers these 2 carbons molecules into the Kreb cycle, and let me put it this way. It delivers these 2 carbon molecules into the Kreb cycle, and CoA is also released, so CoA can go back and bring in more pyruvate into the Kreb cycle like this. So it's released back into. Just kind of erase this over here. Okie dokie. So now this molecule is going to enter the Kreb cycle like this, and coenzyme A is going to be regenerated over here, so it can keep cycling like this. Okay. Now, as I have suggested before, that the majority of the ATP is generated in the electron transport chain. Now, if you appreciate it, we said that these are electron taxis. The NADHs are electron taxis, so they're carriers of electrons. So what do they do with those electrons? What they do is they come here to the inner mitochondrial membrane, okay, and they deliver their electrons to the inner mitochondrial membrane. Okay, like this. Now, embedded in the inner mitochondrial membrane are proteins, okay? And these proteins, the transmembrane proteins, constitute the electron transport chain. So these proteins, all of them in a sequential proteins, okay, they make up to altogether this electron transport chain. What does that mean? Electron transport chain, as the name implies, okay, is a series of proteins which take up the electrons from the electron donors, from all of these guys, and these are high energy electrons, by the way, right? Those are high energy, and they use the energy, and they kind of hot potato the energy, similar to photosynthesis, and they use that to generate, lo and behold, same as the hydrogen ion gradient. So they generate a hydrogen ion gradient. Okay, so they use the energy in the electrons. Okay, the process is very similar to what happens in photosynthesis, and a hydrogen ion gradient is generated. So the hydrogen ion gradient is the end result of production of all of these carriers, electron carriers. And it is this hydrogen ion gradient, okay, it is this hydrogen ion gradient that produces the bulk of ATP, okay? It produces bulk of ATP. Let's go this way. So it produces bulk of ATP. How? Now, we have seen that before using the transmembrane protein complex ATP synthase. Alright, so ATP synthase, if you recall, was a funky looking transmembrane protein that kind of like this. We head over there, and what it did was it generated ATP from using this transmembrane protein, transmembrane hydrogen ion gradient from ADP to ATP. Okay, so ADP plus phosphate group attaches to make ATP. In fact, from these you generate some like 30 ATPs during this process. Okay, so this is a summary of cellular respiration. Okay, let's just go over this individual steps again. So the first step is glycolysis, which occurs in the cytosol, conversion of glucose molecules to 2 pyruvate molecules. This process initially requires sacrifice of 2 ATPs. It's an energy requiring process, but then it gives you 4 ATPs total, so it actually gives you 2 ATPs, which is pittance compared to what electron transport chain gives you, right? So nevertheless, we're going to be appreciative. So for 2 ATP net, it generates NADH, which is an electron carrier. Where is that NADH going to go? It's going to go over here to the electron transport chain. So this is the electron transport chain in the inner mitochondrial membrane right there. So now the pyruvate is generated in the cytosol, again goes into the mitochondria, and then into the mitochondria, pyruvate is converted to acetyl-CoA. This is the second step. This is this enzyme requires a coenzyme A, and generates acetyl-CoA. Acetyl-CoA then goes into the Kreb cycle or citric acid cycle, which further metabolizes this and generates further energy rich compounds, ATP, GTP, and electron carriers, NADH and FADH2. All of these electron carriers then go and produce this transmembrane hydrogen ion gradient, which is used by ATP synthase to generate ATP. Okay, so this is a summary of cellular respiration, glycolysis, cellular respiration. Step 1, glycolysis. Step 2, conversion of pyruvate. Step 3 is the Krebs cycle, and step 4 is the electron transport chain. So that's a summary of the whole process. Inshallah, next time we shall go over this again in a little bit more detail. Until then, as-salatu wa-salamu ala rasulillah wa ala alihi wa sahbihi. Assalamu alaikum.